Hello everyone, my name is Alper Yilmaz, and today I'm a graduate student at Bilkent University in Turkey, and today I will be presenting our recent work on a copy number variation caller designed for ancient genomes, which we have named LYCEUM. Before I dive into the details of our model, first I would like to mention why studying ancient genomes matter. I will focus on three key reasons: origins of disease susceptibility, evolutionary insight, and population history. First, the ancient DNA help us trace the origins of genetic disease susceptibility. It reveals when and where the disease-associated variant first appeared, helping us understand how these risks evolved over time. Today, we know that some diseases affect certain populations more than the others. MS, or multiple sclerosis, is one of those. It's an autoimmune disease, and we know that it disproportionately affects the people of Northern European ancestry. To answer where that risk comes from, we need to look at the genomes of ancient people. A study published in Nature in 2002 and 2004 actually did this. They analyzed over 1,700 ancient genomes, and they found out that the main MS risk variant became common in Steppe pastoralist group in around 5,000 years ago. They suggest that this variant may have helped the immune system to adapt to tough and fast-changing environments faced by those groups. But on the other side, it's linked to the higher MS risk. And secondly, ancient genomes offer powerful evolutionary insight. It helps us understand how Homo sapiens adapt to new environments, different diets, unfamiliar diseases, and even new mental demands as we spread across the globe. Fujians are a good example in this context. These Fujians are ancient indigenous populations from Tierra del Fuego at the southern tip of South America. These people are very interesting because they live in an extremely cold condition, yet they wore minimal clothing, they swam naked in the ice water, and they even slept in the open. A recent study analyzed the genomes of Fujians, and they found out that these people actually carry some genetic variants associated with the heat genetic fat tissue, and this specialized tissue make them endure the region's extreme cold condition. And finally, ancient genomes can reveal the deep structure of human history. It allows us to trace migrations, admixture events, and evolutionary changes. It helps us understand not only where ancestors come from, but how their movement and interactions shape the genetic landscape we see in populations today. Here you see some related studies here. Okay, all in all, ancient genomes, as I said, offer powerful insight to the origin of disease susceptibility, evolutionary insight, and population history. However, the most current findings are based primarily on the SNPs, single nucleotide polymorphism, which represents only a portion of our genetic complexity. So if you want richer and more complete picture, we must go beyond SNPs and begin systematically analyzing the structural variants, especially copy number variations, CMVs. CMVs are large-scale structural variants in the genome that can affect gene dosage, disrupt coding regions, and change regulatory landscape. They are known to influence traits such as brain development, immune response, and metabolism, and many other traits, of course. And CMVs, copy number variations, are often more functionally impactful than the individual SNPs. Yet, in the ancient DNA research, copy number variations remain largely unexplored, primarily because ancient genome sequence data is extremely noisy. The read depth signal, which is required for which is the primary input for the CMV detection, is often distorted, sparse, and unreliable. There are three major factors contributing to this: contamination, chemical and environmental damage, and low sequence coverage. First, ancient DNA is often heavily contaminated with the foreign material. This includes DNA from modern humans introduced during handling or excavation, and microbial DNA that infiltrates the remains over thousands of years. And these contaminants can overshadow the endogenous DNA and mislead the downstream analysis. In addition to contamination, ancient samples are also damaged by time and environment, making analysis even harder. A key issue is fragmentation, often caused by depurination, which breaks DNA into the short pieces and making them harder to map. Another common problem is cytosine deamination, which leads to C to T or G to E sequencing errors, especially at the fragment ends. And also heat, humidity, and other environmental factors speed up this damage and often leaving only a small error-prone portion of usable DNA. And finally, last ancient genomes are sequenced at very low coverage. Here you see the visual comparison between the modern whole genome sequencing sample at the top and the ancient genome at the bottom, aligned with the same genomic region. In the modern sample, you can clearly see dense and uniform read alignments, but in the ancient sample, the coverage is extremely sparse, uneven, and highly fragmented. This is what we typically see in ancient genomes. So in this kind of data signal, even a small fluctuations can be misleading. Okay, so far we have seen that ancient whole genome sequence data is extremely noisy. And this noise limits our ability to detect CMVs using traditional methods. And the question is, can we learn to work with that noise? Can we train a model that recognizes patterns from the noisy read depth signal and still accurately call CMVs? This idea isn't entirely new, actually. In fact, our lab recently developed a CMV caller tool called ECOL, which is published in Nature Communication. ECOL successfully learned to call CMVs from noisy exome sequencing data, which is itself fragmented and variable in coverage. So ECOL showed us that it's possible to use transformer-based method to recognize the CMV patterns from challenging data sets. And that success led us to wonder, could a similar approach work for the ancient samples as well, even under ultra-low coverage and high fragmented ancient samples? But the main problem is that although modern samples are abundant, ancient samples with a moderate coverage are sparse. So we are facing two simultaneous challenges. One, we have very limited number of training samples, and two, our data is extremely noisy. To solve this problem, we first train a generalist CMV calling model using high-quality modern VGS data. For this, we use 550 VGS samples from 1000 Genome Project, each sequenced at around 30x coverage. This gives us a clean, high-resolution read depth signals, which is ideal for training. And our ground truth labels are CMV calls generated by the modern germline CMV caller tools, which produce high, accurate, and reliable CMV annotations from modern VGS data. Note that in this step, we don't use ancient DNA. We only use modern VGS samples because the main purpose in this step is to teach LYCEUM to learn the basic relationship between the read depth signals and the CMV states. And this gives us a generalist model which knows how to call CMVs on optimal conditions. After pre-training our model, pre-training the model with a modern, high-coverage, high-quality genomes, the next step is to adapt it to the ancient DNA conditions, and that's what we do in step 2. Here, we fine-tune the pre-trained LYCEUM model using the data that mimics the challenges we expect in real ancient genomes: low coverage, noise, and degradation. We use two sources of data for this. We use 30 moderate coverage ancient samples here. Each sequence more than 9x, which is crucial because we can trust the CMV calls we make on them. And also we downsample each sample to 5 different coverage levels, and it gives us 78 samples in total. And also we generate simulated ancient samples in 4 different coverage levels. For this purpose, we use Gargamel tool. Gargamel tool allows us to create synthetic read depth signals that includes realistic DNA damage pattern. Yes, all in all, now we end up with a diverse and challenging data sets. But now we know the ground truth because it's either coming from the moderate coverage samples or the fully simulated samples. And after fine-tuning our model, we convert our general CMV model to the specialized model, which now knows how to call CMVs on ancient samples. Here you see our architecture. As I said, our input data is the read depth signal coming from the exon regions, and we embed this read depth signals using two sequential 1D convolution layers here. Then after that, we concatenate this vector with the chromosome-specific token, which provides chromosome-level contextual information. Then we summed this resulting vector with the positional encoding, and the final vector is passed through the cascade of 3 transformer encoder blocks. Then we extract chromosome-specific token and give it to the feed-forward network, and this gives us the probability for each CMV states. To evaluate LYCEUM, we test in two distinct data sets. On the left, we show real ancient whole genome sequence VGS data. We use 7 moderately covered ancient genomes along with 5 downsample versions for a total of 42 samples. And on the right, we include 40 simulated genomes with 10 samples at each for different coverage levels. These are the results of our benchmark. This figure shows the deletion performance on the left and duplication performance on the right. On both plots, the y-axis represents the F1 score, and x-axis shows the sequence coverage levels, starting from the original high coverage to lower coverage levels. We compare LYCEUM against 4 widely used CMV callers: CMVinator, Conga, ContourFreq, and GATK. Among these, only Conga is specifically designed for ancient samples. It's a rule-based method. And we assume there is the CMVinator is the semi-ground truth. And we observe that the LYCEUM shown in orange outperform other tools at low coverage levels. Here you see that it achieves 20% improvement for the deletion F1 score and 39 F1 score improvement for the duplication case over the next best method. Also, we analyze its performance on the simulated samples, simulated ancient DNA samples. Here you see our results again. So in this case, our model outperforms all other tools at every coverage levels for the simulated samples. So it highlights that LYCEUM's robustness under extremely noisy and fragmented condition, which is typical of ancient DNA. We also tested LYCEUM's performance on ultra-low coverage levels. On the left, you can see the results for deletions, and on the right, duplications. The blue line shows precision, orange is recall, and green represents the F1 score. As you can see, the performance remains stable, fairly stable down to 0.05x, but it drops off significantly below that point. Yes. So as many of you know, GC content can also introduce significant bias in read depth signals, which often impacts CMV coding accuracy. We evaluated the effect of GC content on the LYCEUM performance. The plot shows deletion and duplication performance across different GC content bins with different deletions on top and with deletions on top and duplications on the bottom. Each bar is broken down by false negatives in green, true positives in blue, and false positives in orange. And we observed that LYCEUM maintains stable performance across different GC content levels. And this shows that LYCEUM can handle the GC-related read depth changes very well, which is especially useful for working with the degraded and ancient DNA. And to assess the reliability of LYCEUM, we also analyzed the exon-level deletion codes across 50 low-coverage ancient DNA samples from different parts of the world. You can see the origin of all samples in figure A. It's colored from blue, Western Europe, to red, Eastern Asia. And figure B shows the PCA plot based on deletion codes made by LYCEUM. Again, we observed that although there are some mistakes, most of the European and Asian samples are clustered together. It shows that LYCEUM effectively captured the population-level variation and structure through its CMV predictions. And we also performed hierarchical clustering analysis. This figure shows the dendrogram generated using deletion events in the same 50 ancient samples detected by LYCEUM. Samples are grouped based on Manhattan distance and varsity2 linkage. And we observed that LYCEUM achieves clear separation between the European and Asian samples with accuracy of 70%. And finally, we analyze if the LYCEUM deletion calls are consistent with the expected evolutionary pattern. And we expect rare and small deletions being more prevalent. Figure A shows the histogram of the mean allele frequency. And notice that the vast majority of calls have low allele frequencies. In other words, rare deletions are much more common, which is good in our case. And the figure B shows deletion size distribution. And we observed that short deletions are much more prevalent. This is also consistent with what we know, because large deletions are more likely to be deleterious and therefore less common. So this analysis shows that LYCEUM is not only a technically sound tool, but also biologically meaningful. To summarize, LYCEUM is the first deep learning-based CMV caller tailored for ancient genomes. It's designed to tackle key challenges in ancient DNA analysis, including contamination, low coverage, and noisy read depth signals. And LYCEUM outperforms existing CMV callers at very low coverage levels. And the CMVs called by LYCEUM correlate with the demographic history and evolutionary pattern in ancient population. And I would also thank the ISMB HiTSeq community for the travel fellowship, which made it possible for us to present this work here. And also big thanks to our team. And you can access our code and datasets in the links. Thank you.
Thank you. Do we have any questions? I think Francisco has one.
Yeah, sorry, I maybe missed it, but do you tested recall a different from both your simulated and real data at different lengths of events?
This one, simulated.
Yeah, but this is the overall. I wonder if longer events are difficult than shorter events to call, right? Because I will imagine that in low coverage situations, it's going to be easier to detect large events versus small events. So I wanted to know, what's the trade-off there? Are you mostly finding large events and the small events you cannot see? If we know that, in fact, small events should be more frequent because as they become larger, they are more deleterious. So I think that would be good to see what's the distribution there.
Okay, so we actually, our model calls exome-wise calls. We don't give the breakpoints, and we also didn't analyze its performance on the different event sizes. So, but we will do it in the future, maybe.
Okay. Do you have another question? Okay.
Thanks. We have time, right?
Yes.
Okay, so that's
2 minutes.
Thank you. Just wondering, so how it's a little bit puzzling that in the PCA you have some kind of that we know Europeans cluster to Asian signal and the other way around. Wondering, so this is like extremely low coverage stuff, but if you were to use only the ones that we can normally use, like 10x coverage or so, how does it look like, the PCA? Like if you were to do the exact same thing, but with more common colors?
Actually, we only tried with the ancient samples with the low coverage levels. We didn't try with the high or moderate coverage levels. We tried, but we did not visualize this in PCA plot.
My question here is because whether the ones that are missed place are actually the ones with the lowest coverage, like if there is an effect on that.
It can affect, but I'm not sure right now. Thank you.
Thank you.
Okay, one more question, Anna.
Yeah, this is not a question, it's just a comment. I don't know if you saw, but recently it was sequenced the first whole genome information of a sample of the ancient Egypt. So it might be super interesting, like at some point, if you can do this application implementation into this sample.
Okay,
Yeah.
yeah. I will look into it. Thank you.
Yeah, okay, go ahead.
I was wondering how hard it is to retrain the model for other organisms,
Other organisms?
non-human.
Actually, we share our model with the GitHub link. Maybe you can download it and you can train our model with your samples as well. So it's not so hard. Maybe we can talk later after the presentation. Yeah.
Thanks.
Thank you.
Okay,
Thank you.
thank you so much again.